Pino y al doctor Encinas a subir a la mesa. Bueno, seguimos avanzando en la tarde, seguimos intentando generar ideas, proyectos y exponer ideas de investigación que pudieran ser útiles en el campo de la epilepsia refractaria. Y bueno, yo siempre, no sé cómo presentarles. A la doctora del Pino viene de Valencia y el doctor Encinas viene de aquí, de la Chúcarro de Vizcaya. Yo creo que tienen una cosa en común, que los dos fueron muy osados de atenderme en unas situaciones curiosas, ¿no? Porque a Isabel hace muy poquito, media hora casi, de un bombardeo de ideas en el Centro de Investigación Príncipe en Valencia, me dijo a todo que sí y aquí seguimos andando colaborando. Y hace mucho tiempo, no sé lo que pensó Juan Manuel, que le escribió una persona y se acercó allí a un workshop de investigadores y estuvimos una hora hablando sobre las posibilidades de su investigación en el Dravet y aquí seguimos. Por lo tanto, aparte de desarrollar su trabajo de la forma que lo hacen, son lo suficientemente osados para decir que sí, para abrir puertas a veces a locuras, ¿no? Y le dejamos con Isabel para la primera ponencia.
Buenas tardes. A ver, yo soy una investigadora española, pero voy a ser la primera que va a calentar un poco el plan del tema del inglés. Y la razón es que yo siempre, desde que empecé el doctorado, me fui fuera, entonces mi carrera investigadora ha ocurrido siempre en el extranjero. Mi cerebro piensa en inglés. Y aunque estoy entrenando mi plasticidad para poder pues venir a este tipo de conferencias y poder expresarme en mi idioma incluso, me resulta aún un poco difícil. Entonces, prometo que si vuelvo, lo haré en castellano. A ver, so I'll switch now to English, and I will start by saying that I'm a developmental neurobiologist. That means that I'm interested in understanding how the brain is formed in mammals. And today I'm going to try to convince you that why this is important and why this is relevant to understand neurodevelopmental disease. When I talk about neurodevelopmental disease, sorry. Now it's good? Okay, when I talk about neurodevelopmental disease, that spans from epilepsy to autism to schizophrenia. So today I'm going to tell you a little bit of what we have learned about all of these disorders, about learning the alterations and the changes that all of these disorders have in common or have differentially. I will start by summarizing what we know and what we learned from the perspective of developmental neurobiology. The genetic studies have allowed us to identify genetic risk factors predisposing to different neurodevelopmental disorders. And actually, Dravet is a very good example of that, because the genetic predisposition of the SCH1A, it's very, very strongly contributing to developing epilepsy from the spectrum of Dravet syndrome. However, we also know that the genetic risk to develop neurodevelopmental disorders, and in particular epilepsy, is the genetic predisposition is highly polygenic. That means that if you are lucky, you could have a monogenic disorder, so one gene contributing to the disease, but in many other cases, you have a polygenic risk. That means that there are multiple genetic factors that contribute to the disorders that are not known. That's happening for some cases of epilepsy, for some cases of Dravet, but also it's very common in autism, for example. So what we propose is, I think, an original perspective of how to approach disease. If traditionally, what we are doing in our mouse models, because that's why I would try to convince you why mouse models are important, is that we are able with the mouse to recapitulate, the mouse is able to recapitulate some behavioral phenotypes that are reminiscent of neuropsychiatric or neurological symptoms. In the case of epilepsy, it's very clear that to identify the seizures, but also some associated cognitive deficits that we can measure in the mouse very easily during behavioral tests. And this has allowed us to develop and test the possible treatments, right? But we believe that many of these symptoms can be assigned to changes in the developmental trajectory of the formation of the brain. Depending on when the development of the brain is altered, we could somehow predict whether disease is going to lead to autism, schizophrenia, or it's very clear that early on leads to epilepsy. So what we are trying to do is to identify functional changes at different biological scales that can help us predict or assess that brain during the development is gonna lead to epilepsy or to other neurodevelopmental disorders. So in the mouse is a very useful tool because we can use these kind of genes that are very relevant or very strongly associated to Dravet or to other kind of epilepsy or encephalopathies, and with these models, we can assess whether there are functional changes at the molecular. We can identify which is the neural substrate that is majorly impaired, because not all the brain is the same. The brain is composed of many different neural circuits. We can now narrow down which are the cellular as well as neural circuit targets that we should timely target. Just to give an overview of why and I would like to remark why development is so important, think as neurodevelopment as a decathlon, as a triathlon of so many different steps. You have a first block of neurogenesis that is super important to make the brain grow, followed by synaptogenesis, axon growth, myelination, and synaptic pruning. This is the time course in the human, right? And this is just a simple perspective or simple example of what we are, currently trying to model in mice. We are constantly updating this developmental sequence of process, and we know it's very relevant because for some of these specific processes that are happening during development, if you impair them, you have a consequence that it might be more or less relevant for adult brain function. So our goal in the past years has been, in general, as a field in the developmental biology, but not of so many groups, has been to understand how many of these basic processes and when are they impacting on adult brain function. We have done this with different neurons, different time points, etc. We still have a lot to do. This developmental sequence that we are trying to relate to epilepsy or to other neurodevelopmental disorders is characterized by a sequence of critical milestones, what we call the critical periods in neural circuit development. We know, for example, I could give you many examples, but just have a look, for example, at the GABA function that is very related to epilepsy, and in this case to Dravet syndrome, because we know the main genetic risk factor is associated to GABAergic interneuron function. You have a shift from depolarizing to hyperpolarizing effect of GABA, and this is very acutely changing at birth, just to make you aware of how many things are going on and how what we know as adult circuits are changing in development. There is a huge plasticity, there are huge changes, and if we if we are lucky enough to know all these different biological scales in the adult brain or the almost pre-adult state in mice or in humans, imagine that we need to know all of these different characteristics of the brain at very critical time periods during development to understand neurodevelopmental disease. So at the end, what we want and what we need is to deconstruct brain development in normal conditions and specifically in each pathology of neurodevelopment, such as Dravet syndrome. And to do this, we think that the mouse brain is very good model because we know a lot about the developmental milestones, the critical periods happening in the cerebral cortex. Actually, these milestones in the developmental sequences are different depending on the region of the brain. They follow a gradient of caudal, rostral, mediolateral. So you have complex cortex development. I think is the most complex structure that we can study. But we think that by assessing and dissecting these developmental sequences, we can find early signs or early biomarkers that can lead us to understand better and to diagnose better disease. Why I say this is because when we talk about why you are using this or this other treatment or checking it preclinically, your compound, the common answer is I check in adult or in pre-adult state because Dravet will never be diagnosed until seizures appear. And what my goal and our goal from the developmental biology is to challenge this view and to try to make that people try to identify what's going on before seizures are happening. We think that by understanding development, we can do that. So the first approach that we use in order to understand the development of the cerebral cortex early on and readouts that can help us identify these early changes is the functional behavior of cortical networks early after birth. So in the mouse, if you look at the first week after development from P0 to P7, so P means postnatal day after birth. So if you look at this particular time window and you try to assess cortical network organization with calcium imaging, because when neurons are active, there is an entry of calcium, and with calcium indicators, you can see whether a neuron is active or not. So when you measure this fluorescence, I hope... no. Can we play the video from... here? Sí, es que no puedo. Okay, we tried it before, it was working. Now it's not working. Well, no. The videos are there, but it's not. Okay, so wherever. Now, yes, okay. So when you measure, you try to look at okay, it's not possible. If I would have the opportunity to see the video, you will see that during early stages after birth, you have a very sparse activation of neurons at the P0 that you can see by a blinking of very few neurons, while at P7 or P5 to P8, you can see that neurons synchronize and fire in a more coherent way. So that means that the synchronization of cortical networks evolves in this particular time window of 7 to 8 days in the mouse neocortex. This has been characterized by the team of Rosa Cosar, which were the first showing this in ex vivo. Now it's working. Okay, so do you see the, I guess you can see, even if you are not familiar with it, that the activation of neurons is very sparse and not very coordinated, while at P8 you have these waves of synchronization, right? That was pretty fascinating. So, moreover, if you look at the dynamics of these cortical networks, you can see that they follow a very precise patterns that I'm not going to go into detail, but they go from single spikes of few neurons, every single dot is a neuron, to coordinated activation or deactivation of cells that evolves from plateau assemblies, because they are very long, here in red, to early network events, etc., GDPs. Very important also to the formation of the synaptic connectivity of neural networks. This is not only important to understand the dynamics, but to know also how this, the cerebral cortex is forming networks, connecting with each other, how the neurons connect across this first postnatal window. And we actually know that, for example, the plateau assemblies are driven by gap junctions, which are like connectors through the membranes of neurons with each other, while glutamate and GABA, which are two neurotransmitters, are leading to different network oscillations. This is actually also very sensitive to hypoxia, and is very sensitive to systemic changes in hormones and environmental factors. So what we wanted to address, or what I would like to propose, together with people in my institute, is to address this kind of to address neurodevelopment in Drabe. And as an example, I will tell you what we have done with the NA2F model of West syndrome. This NA2F is a factor that it's a nuclear receptor with a known ligand, that's why it's called orphan nuclear receptor. We don't know how it's activated, but we know it's very important for the neurodevelopment. We know that it predisposes to West syndrome plus other syndromes, and which is an early onset epileptic encephalopathy. So our question was, how can we identify early readouts of developmental abnormalities in this mouse model in the cerebral cortex? To do this, we employed calcium imaging with these calcium indicators, similar calcium indicators that I have shown you. And we did this within this, everything we have done is within this early time window after development from P0 to P8. And we observed that this is just a schematic of the measurements of calcium, very basic, not really comparing which kind of oscillator activity, but what we observe is that the activity of single neurons at P1, here in red, in the mutant was very highly, significantly higher compared to control in calcium bursts, while during development it was normalized. However, when we see that the activity of neural networks is normalized, when we assess for this synchronization, whether all neurons are active at the same time or not, we saw that this synchronization is decreased. We suggested that even in mouse models where we are recapitulating mutations that have to do with West syndrome, we can find early cortical network alterations measured at the level of functional organization. To understand why this is happening in any kind of disorder that has to do with the cerebral cortex activity, hypersynchronization, ictal activity, preictal activity, all of this has to do with two major factors. First is the connectivity between the neurons, and second is the excitability of the neurons. And in this meeting, you have many times heard about the voltage-gated sodium channel that predisposes to disease. All of these voltage-gated ion channels are determining the excitability of neurons that look, that if you test them, look a little bit like this. You have the action potentials that Sarrans was talking about and was explaining. So what we do is to understand how the excitability is impaired early on in development. Because actually, this pattern of excitability you can see here in control and you can see here in mutant is not the same across development. It follows a very, very strong change and very strong maturation. So it's important to understand how is the intrinsic excitability of neurons such as this very early on, in our opinions. So that's what we did. We assessed how pyramidal neurons in the neocortex are which is the excitability of these neurons early on in development. So you can see that even though they look pretty similar, you have very, very subtle changes in some currents. For example, in this hyperpolarization activated current, you see here this, what we call the SAC, that is reduced. Why is that important? It's because every type of current, even if you have changes in action potential with that you think it's very small, but it can be measured precisely, and it's very important. All of this is determined by ion channels, and we can assess which kind of ion channel we are lacking, even if we don't know, like in the case of the Na2F. When we measure this, we see that the SAC is decreased, and of course, we want to know which are the ion channels that are, or validate that which are the ion channels that are changing. For this, we do molecular approaches such as qPCR. We take brains very early on from the cortex. We do, we assess the transcriptional, 3 minutos. We assess the transcriptional levels of ion channels, but Western blot we triple validate that these ion channels are changing, and one of them that was strongly changing was HCN1. Here you can see a strong reduction. You also have changes in other potassium channels, also NMDA receptor, which you can also observe in Western blot. So the question is, do we have specific ion channels changing and others changing due to compensation? That's another problem we face. Okay, so what we do is to find out whether our gene of interest is directly regulating ion channel expression. For that, we do some promoter sequence analysis to understand whether our gene of interest is binding to the promoter that is driving the expression of the ion channels. And from the ones that we saw that they were changing, we found that HCN1, for example, is directly regulated. Here you can see the quantifications very strongly, which suggested that indeed voltage-gated ion channels were changing in our model where we didn't know what is the functional change that was going on. In the future, what we are putting in the pipeline is to do the single-cell mRNA sequence. That is an approach that you use to understand the whole transcriptomic profile. In normal words, you want to know the whole of the cell, what is changing, ion channels, if possible, signaling cascades, and this can be done with different approaches. One of them is the patch-seq, they call it. Is you patch a neuron when you are doing the recording, and when you finish the patch, you retrieve the cytosol. And with the cytosol, you are taking all the mRNA, you hope it's a big bunch of mRNA within the cytosol that you can see here. You can take or not the nucleus if you're lucky. And when you have all these samples, you just amplify, you sequence it, and you can get even up to 2,000 targets using this approach, where you can see the whole landscape of your molecular changes. And I think that's super relevant to understand disease, wherever it is, even more in development where we still don't know too much about. So in summary, what I wanted to bring today is the idea that neurodevelopmental disorders such as West syndrome or Dravet are appearing due to a sequence of developmental events. It is important to understand these developmental events, and for that the mouse is super useful. And in the case of West syndrome, we found that there are alterations early on after birth in network activity that we think they are due to intrinsic excitability directly regulated by this Na2F mutation. So in general, yeah, we think these approaches can help us not only to find early biomarkers, functional changes that can help us understand whether cortex is going in the right direction or very early on is going in the wrong way. We also think that this is also super good to identify time windows where we can put our treatments in place in order to be able to do the earliest intervention possible, and which we think has many more potential due to the ability of the brain to plastically react then or retake a normal development. And with this, I conclude. I would like to thank, well, I started this work in Bordeaux, together with the lab of Xavier Leine-Kugel and Michel Studa's lab, that she's It's amazing, and we are very excited to start work with Ivo and with Jan Tonnesen, that I hope helps us to move towards the calcium imaging in vivo. And yeah, this is my small group that I just started in the beautiful city of Valencia, and thank you very much for the attention.